Hey guys, welcome to Unit 5. This one is on inheritance, and this one doesn't have like one of those neat, snappy little phrases that we're going to be able to do, like differential gene expression or differential reproductive success. This one is the central dogma, Mendel protein synthesis. Now, you probably have heard central dogma, but you might not remember it. You definitely remember Mendel. You've heard of protein synthesis, but it's very likely you don't really understand what protein synthesis really is and what the implications are for it. So we're going to spend some time on that today. And remember that protein synthesis is a conserved core process, one of those processes which is universal among living things. So here is a shocking phrase for you. DNA is the primary source of hereditary information. And occasionally RNA is, but we spent a long time trying to figure out what the hereditary information was stored in. And we're going to tell you some of the story in this unit behind that, because it is a great background for understanding the methods of science, science processes. And there's also some stuff in there about Watson and Crick that make them look not quite so good in relationship to what we would expect to see in ethical behavior. So it's kind of a fascinating story. But here we have a bacterial chromosome right here, and here we have a eukaryotic chromosome. Eukaryotic chromosomes, you probably know, are linear in shape. Bacterial chromosomes are rounded. And then here is a really cool picture of a bacterial chromosome, how it actually looks. And it's not really a circle. It's kind of packed in there, wrapped around some protein so we can get to it. And the eukaryotic chromosome is wrapped very tightly around these other kinds of proteins called histones. We'll talk about the structure of those a little bit later. So anyway, you know about chromosomes. You know DNA is hereditary material. You know bacteria, viruses, bacteria have different structure to their chromosomes than eukaryotes do. And we also know that viruses can have either DNA or RNA as their hereditary material. All right, so in that fascinating history I was talking about, for discovering that DNA was a hereditary material, probably there's several scientists that you're going to need to know. Hershey and Chase is just one of them. And Hershey and Chase did this really cool thing where they took those viruses that infect bacteria called phages, and they gave them a radioactive protein. And then they took them and they put them into a simple blender, stuck them in to mix them, stuck them in the centrifuge afterwards, and tried to see if they found any proteins in them. And what they found was that the proteins did not pass on hereditary information, but if they did the exact same thing with radioactive DNA, that the DNA did pass on hereditary information. So at this point right now, we're pretty sure it's DNA that's hereditary information, not proteins. So that's Hershey and Chase. Some other stuff that they found out was that DNA was a polynucleotide, and a polynucleotide, of course, is poly means many, nucleotides are the basic structures. We'll show those in just a second, of nucleic acids. So DNA is a long string of nucleotides. And then finally, a guy named Erwin Chargaff came up with these rules. He said, this is kind of weird, but the percentage of A's and T's always equal each other. And so A's and T's equal each other, and then C's and G's also equal each other. So we had that kind of information about DNA. One is that the bacteria got incorporated into the DNA, but did not get incorporated into the proteins. And second was that the DNA was this long chain molecule. And third is that the A's and the T's had the same percentage, and C's and G's had the same percentage. Now, this is a kind of a little bit of a tragic story, but Rosalind Franklin was a pioneer in a process called X-ray crystallography, X-ray diffraction, excuse me. And they would take pictures of DNA that was both dry and wet and try and get these X-ray pictures of them. And this particular diffraction pattern was taken by the leader of her lab, Maurice Wilkins, and shared with Watson and Crick. And along with the other basic information that they had, they were able to put C and G together, and then also A and T together, so they knew that DNA was a polynucleotide, that A's and T's equaled each other, C's and G's equaled each other, and that C's and G's actually had 3 places where a hydrogen bond could form. A's and T's only had 2, and that allowed them to get the basic information they needed to get the structure of DNA. A couple of other things about it that they knew was that each Nucleotide was 0.34 nanometers. If you took 10 nucleotides in a row, you would get one twist of your DNA. So every 3.4 nanometers, you get a little twist in your double helix. They knew that there were purines and pyrimidines, so that purines have 2 rings, and pyrimidines, like this one right here, and pyrimidines have 1 ring. So a purine like guanine would have to pair with a pyrimidine like cytosine if our ladder was going to be the same width across. So all kinds of little factoids, information about Watson and Crick to come up with the structure of DNA. But the history of Rosalind Franklin, we'll talk about how she contributed, and then we got kind of left out of the mix a little bit later. All right, so once they discovered DNA structure, as they were doing that, in their original paper published in 1953, they said that the structure of DNA also suggested how it's copied or replicated. And you are going to need to know, now they didn't know the names of the molecules that carried out the replication. They just said that it would unzip like a zipper, that the hydrogen bonds that were joining these bases in the middle would break apart easily, but the sugar phosphate bases along the side, like we see right here, would not. And so the DNA could be pulled apart, like we see right here, but that would suggest that the type of replication that was going to be occurring here. And I'll give you a spoiler alert here. That replication is semi-conservative. Semi-conservative is pretty straightforward. Conservative means preserved exactly. So conservative right here would be that we would have the exact same thing that we started with, an exact duplicate copy, a Xerox copy. But we're only semi-conservative. That means that on each new strand of DNA, we have one new strand, which is the light blue in this picture, and we have one old, which is the darker picture. So DNA replication is the semi-conservative. Its process is something you're going to need to know. It is a conserved core process along with protein synthesis that we'll be doing in this unit. And you will need to know the processes, that are, the kind of steps that are going by, the molecules that are important in DNA replication. All right, but it is semi-conservative. Now, we're going to talk about this other super cool molecule called reverse transcriptase. And reverse transcriptase is an evolutionary adaptation provided by viruses. And reverse transcriptase is a protein that we take advantage of in genetic engineering. We'll talk about that a little bit later. But in viruses, what it does is it copies RNA to DNA. And that provides us with a little bit of an indication that viruses themselves, again, they have processes which are not similar to living things. It's not a conserved core process. Not everything has reverse transcriptase. But knowing about reverse transcriptase allows us to take advantage of its abilities. And one of those is if we can get some mRNA or we can get some RNA from a virus, we could copy it into DNA. And it doesn't have to be from a virus. We could copy anything. We get RNA from you. Let's say we wanted to get a particular protein out of you, just take the mRNA out of you, and then we're going to use reverse transcriptase, and that will turn that mRNA into a DNA sequence. DNA polymerase, one of those proteins involved in replication, would then make a second copy of the DNA. So we have this nice little complete strand of DNA. So that's a reverse transcriptase. More on that later. Right, so DNA and RNA have structural similarities, and that helps define their function. So in DNA, we have, in RNA, we have adenine, guanine, cytosine. These are nucleotides. In DNA, we only have thymine. In RNA, we only have uracil. So there's a slight difference in terms of the nucleotides that are present. So 3 are the same in both, 1 is different in both. The sugar is the same in DNA. The phosphate is the same in DNA and RNA. The sugars are different. The sugar is a deoxyribose in DNA and a ribose in RNA. And the third thing about them is that the RNA is single-stranded and the DNA is double-stranded. So those are some structural similarities and differences, and that, again, that helps define our function. One idea I want you to understand is that DNA is very stable. It is among the most stable organic macromolecules, which is why when we pull out fossils, the last thing that's going to be preserved in there probably is not fats, not carbohydrates, not proteins, but nucleic acids. So very stable. RNA, far less stable, but it also has a great deal of flexibility in what it can do. So what exactly does RNA do? Well, we have at least 3 different types of RNA that you know about. One of those is mRNA, or messenger RNA, and that is what is transcribed when we're trying to make a protein. So that takes the information from the DNA and takes it to the ribosome. And the ribosome itself is actually made up of 2 things. It's made up of rRNA, ribosomal RNA, and it's made up of proteins. And the ribosome itself is then assembled, and then it reads the mRNA, and then it takes from a 3rd form of RNA called tRNA transfer RNA, amino acids that the tRNA has picked up. They are then placed on that tRNA by a protein, and then they are brought to the ribosome, where they are assembled into a protein. So RNA's primary job is involved in protein synthesis. We'll talk about that later. But it also is really doing a great job of controlling gene expression, which we'll talk about in the next unit. And a couple of other things I want to introduce to you. This is brand new to you, and something relatively new in biology is what's called RNA interference. And you can kind of see here that there's a couple of different forms of RNA, miRNA or microRNA, and Si, or small interfering RNA. So RNA interference has these 2 different forms of RNA, and they are going to be interfering with translation. They may bind to an mRNA molecule and keep it from getting translated. That would mean that even though the cell has said, hey, I want to make this protein, this type of RNA can come in there and block it. And then it also plays an important role in gene expression. So sometimes it can cause proteins to be produced in higher quantities, which is really cool for us, and it also can block viral proteins from being produced. So if we're looking for a way to protect ourselves from viruses, these RNA interfering molecules might be the thing. So we have rRNA, mRNA, and tRNA, and then we also have miRNA and sRNA. All right, the central dogma. And we've kind of been hinting around at this, but central dogma is pretty straightforward. It says that DNA codes for RNA, which codes for proteins. So the DNA directs the protein synthesis. And there are 2 processes. One of those processes is called transcription, and the other one is called translation. Transcription occurs from DNA to RNA. Translation is when the ribosome reads the RNA and turns it into a polypeptide or a small protein. Lots of polypeptides can be put together in different combinations to make different kinds of proteins. But these 2 babies right here, transcription and translation, that's the sum of protein synthesis. All right? And this is a central dogma. It says that information is flowing from one direction to the other. Like we saw with the reverse transcriptase, occasionally the RNA can code for DNA. That would be kind of breaking up that central dogma. Which is why, as a belief system, biology and science is not a great thing to work at, because, of course, there's always exceptions. All right, so you are going to need to know the processes of transcription and translation. You've already studied those in the past. The details here are going to be exactly the same as what you have seen in the past. If you had it in 7th grade, I know you had it in 9th grade, and it's really nothing changed. It's just a matter of you understanding how it works and being able to explain it to someone else. But knowing how protein synthesis works is a foundational core for understanding proteins. Remember, proteins, like we talked about in the first couple of units, they do all the work. So inside your cell, if you can't make proteins, you're not going to survive. And so understanding that process of making proteins is going to be pretty important. So, all right, so let's give you a quick example here. We've got DNA, that's where information is stored. We know that you can read by transcription the information from DNA, and that's going to get translated into proteins. Those proteins then can control transcription, they can control translation, they are going to control all those metabolic pathways that we have been referring to throughout the year, metabolic pathways for everything in your body. And they can also affect expression of phenotypes. All right? And of course, the environment is going to influence the gene expression, and it's going to influence the types of proteins that are present, and also can influence then, as a result of that, the phenotypes. So the environment could be weather, it could be living organisms, it could be anything that's going to cause or change gene expression. So that's kind of a summary little thing for you about the central role of proteins and DNA in living things. And it's kind of a cool little graphic here. And these are different areas that you could actually study So there are people who are in proteomics and they're studying structures of proteins and how that affects function, and people are studying metabolomics and they're looking at metabolism and the different biochemical pathways and how they're affected by defects in proteins. And of course, genomics is most of the stuff you think about when you think about genes. All right, so once we know that this whole process works, and it works the same way in all living organisms, the great thing is that we can then use engineering principles to take advantage of those processes and then create our own proteins, or create proteins for specific things, or create proteins where proteins are needed, that kind of thing. So one of the things that we're going to work on is gel electrophoresis, and gel electrophoresis is pretty simple. You're basically going to move molecules through a gel, like Jell-O. The gel slows them down, and then we have a charge on both sides, and if they're negatively charged, like DNA is, and you put them at the negative pole, they're going to migrate towards a positive pole. The bigger molecules move farther, or shorter distances, and the small molecules move longer distances. And so gel electrophoresis can separate molecules by size and by charge, and we can use it for crazy stuff like identifying a gene of interest in a normal organism and an abnormal organism, see how they're different. We can identify you as if you were a crime victim, and compare your DNA to suspects and find out which person's DNA was left at the scene of a crime. And then the other thing that we can do with it, once we've isolated the gene of interest, like in this picture right here, we have a bacteria, and the bacteria has its chromosome, and it has what are called plasmids. We can take out the plasmid from the bacteria, we can isolate that, we can expose that to a gene of interest that we have pulled from this gel, and we can put them both together in a little test tube, pour them in the test tube, add a little enzyme in there called ligase. Next thing we know, the gene of interest for the bacteria, the gene of interest from the eukaryote is in the bacteria. And then the bacteria take that up, we grow the bacteria, we give them a nice, funny, great place to live, great temperature, plenty of food, and in return, they start making proteins for us. And they have lots and lots of things that they can be used for, such as human growth hormone, insulin, TPA, which is a drug that dissolves blood clots, and it's a huge lifesaver. So if you never knew anybody who had a heart attack, it's very likely that the medication that they gave them came from bacteria that were grown in not a petri dish, but in a giant container. We isolated that. Can also use to alter the bacteria so that their function is different. Maybe they're eating oil, like we saw in the Gulf disaster in 2010, 2011. And also, we can have a gene of interest inserted into a different organism, so we can make a tobacco plant blow, or we can give it a pesticide resistance. So tons of stuff we can do with engineering. All right then, we know about the cell cycle. You know about mitosis and meiosis. You've definitely studied those. Now we're just going to put those in context here. In eukaryotes, the information is passed on through both mitosis and meiosis, and then the cell cycle is how a cell lives. There are cells which go through the cell cycle very rapidly. From after mitosis is formed here, they may form one new cell, and they may rapidly move through these guys, and then they may undergo mitosis again. Other cells may never go through mitosis, and they may be stuck in a phase that's not called G1 or growth 1, but G0. And those are processes we'll talk about in more detail later, but some cells can undergo mitosis a lot, some cells undergo mitosis rarely, some cells undergo mitosis not at all. And the information that's passed on through that is how they move through heredity. Now, you can study both mitosis and meiosis, or meiosis, and I'm expecting you to understand the processes that are involved, and that mitosis is going to be producing 2 genetically identical cells, while meiosis is going to be producing between 1 and 4 genetically different cells. And the differences between those 2 phases we'll study in a little bit, but you're not going to ask you. I'm not going to say, hey, we're going to spend a lot of time on these. You're going to need to know them. All right, here's our buddy Mendel, and Mendel is going to be one of our seminal figures in biology, and he's going to be greatly aided by a bunch of other guys, namely Thomas Morgan. But you guys know Mendel. You just need to review Mendel and his the question he was trying to answer, which is, why does a trait such as white flowers, appear in one generation, disappear in the next generation, and then reappear in the third generation? That's the question that his research answered. Unfortunately for him, nobody understood it. His work was lost for 40 years, and only later, when we discovered chromosomes and what they do, did we understand how his process of segregation, which is one of his laws, literally was meiosis, and that independent assortment meant that genes that were found on two different chromosomes would assort independently of each other. So Mendel's laws work great within their particular context. They can give us these great ratios that we see here. They're very predictable. And lots of your characters, your traits inherited in humans and other organisms follow Mendel's rules. Right? Unfortunately, Mendel does not extend to every inheritance situation. And Thomas Morgan came along studying fruit flies around the turn of the century to the 1920s, and he found that more than one gene can be found on the same chromosome. If more than one gene is found on the same chromosome, then they are inherited together instead of independently, and that violated one of Mendel's laws. And then the second thing was that would give us different numbers. So we can decide if a inherited trait is Mendel or not Mendel based on the numbers of offspring for a particular cross. Second thing he said was that, hey, sometimes when these non-homologous chromosomes, or homologous chromosomes, get lined up with each other, sometimes they have this particular event that's called crossing over occurring. And he was the guy who identified crossing over. The percentage at which it happened, it's a very rare occurrence, but it does happen. But that is the second thing he did. So one was that more than one gene can be found on chromosomes, and we know today that on human chromosomes we probably have an average chromosome has about 1,000 genes. And the second thing was crossing over, that two homologous chromosomes can cross over. This came from dad in blue, this came from mom in red, and occasionally they may, through a crossing over event, have segments of the chromosomes switch sides. And that gives us new genetic combinations that we did not have before. And of course that changes the inheritance. All right, so that allows us to say that Mendel doesn't explain everything. So Mendel works for what Mendel works for. Mendel doesn't work for everything. If you're a male and you cannot see these numbers easily over here, you cannot see these numbers at all, then you have color blindness. Color blindness is a sex-linked trait. It is found on the X chromosome. Boys have one X chromosome and girls have two X chromosomes. And if the chromosome on the boy is the recessive one, it's going to be expressed. If it's on a girl, she may get two X chromosomes. She's going to get two X chromosomes, and she may get one that's normal and one that is abnormal, or the recessive. In which case, she will not be expressing the sex-linked trait. So she has not color blindness. So color blindness is pretty much exclusively a male event, and that is because they are found on a chromosome. And as a result of that, it's found on a specific chromosome, which is the X chromosome. And so Mendel explains some stuff, and some stuff is not explained by Mendel. They're two systems that are kind of match up with each other. So it's either Mendel or it's not Mendel. All right. So that's a quick summary from this unit, and that's all we have for you today.